Hi guys, Mrs. Vance again. This is the second part of notes for Unit 7, which is about DNA replication, or how DNA manages to make a copy of itself. This is a kind of a complex process, and so I'm going to actually stop a couple of times in here and show you some short videos that'll kind of help illustrate some of the things we're discussing. But it's really interesting the way it works and the fact that it works so well that it happens all the time. So DNA replication is going to depend upon that base pairing that we talked about the last time. Base pairing, remember, A always pairs with T and C always pairs with G. Those are called complementary base pairs. When Watson and Crick came up with their idea for the way the DNA was constructed, they one thing that they noticed was that this kind of made sense in that it showed how it might possibly be copied. Okay. And there were a couple of different ideas about it, but it was shown that their original idea was actually the correct one as things were tested out later on. So basically, it's called a semi-conservative model, okay? Meaning that you have the two strands of DNA. They're going to separate from each other. Each strand is used as a pattern or a template to produce a complementary strand using that base pairing that we talked about, A with T and C with G. And so if we take the left-hand piece, then we put all the complementary base pairs to pair with that one on the left, and we take the right-hand piece and we put complementary bases to pair with the ones on the right, and then you have two identical strands, each composed of one original strand and a new strand that was replicated in the process. So we'll look at how that works in just a minute. Okay, so here's our parental molecule. That's what we call the parental molecule, the first one. So first of all, we're going to separate the strands. They're going to each serve as a template, and then free nucleotides in the cell can be used to pair up with their complementary bases on each side. So each side is serving as a template to make the other side, and what you end up with is two identical strands. Notice the order of the bases is the same on both sides. Each one is one original strand and one new strand that's composed in the process of replication. So you end up with two identical molecules of DNA. Why is that important? Well, every cell in your body needs to have a full set of all the instructions, a full set of DNA. And in order to make sure that every cell has what it needs when cell division occurs, before cell division occurs, the DNA has to be copied so that each daughter cell that forms in the cell division process will have its own full copy of the DNA, and it has to be copied exactly so that the cells have exactly the same information to do what needs to be done. Here's another model. This shows the ribbon model once again, showing that what happens basically is the strand starts to unwind, and as it unwinds, the blue strand is the original strand. Then the complementary bases in gray are placed in the position with their complementary base pairs, C with G and T with A, on each side, so that you end up with two identical molecules, each half old and half new. Now, the interesting thing about this is that it happens in two directions at a lot of different places at the same time. If you took all of the DNA in each one of your cells and stretched it out full length, you would see that each one of your cells, and you have trillions of cells, each one of your cells has about 2 meters or about 6 feet of DNA. If you stretched it out end to end, it doesn't work that way because you have different molecules, but the point is, if you added it all together, it's really, really long. So it's a lot of work, and you've got billions of base pairs in every cell. And so it would take a long time if you just started at one end and went all the way down to the other. And so actually DNA replication occurs at multiple sites at the same time. The replication occurs in the 5' to 3' direction. Remember when we talked about the positions of the carbons in the sugar molecule? Is that it's important for you to know which one is which. And so we start with the oxygen, and we go clockwise from there. And so the number 1 carbon is where the base attaches. The number 2 carbon is the bottom. Number 3 carbon attaches to the phosphate of the previous nucleotide. The number 4 carbon is out on the side, and the number 5 carbon attaches to the phosphate, which is going to attach to the next nucleotide as you go up. So since your two sides of the molecule are anti-parallel, what happens is that on one side, on the 3' to 5' template, in other words, with the number 3 carbon at the bottom and number 5 at the top, replication is going to be continuous in that direction. We're going to add a complementary base to a complementary nucleotide to each one starting from that end. On the other side, though, we can only add from a nucleotide with 5' position to the 3' carbon, and so it's going to be discontinuous on the other side, forming short segments that will later then be pasted or glued together. Let's watch this short video that shows how this works. This is a simulation, of course. This is not an actual video of what happens, but it gives you an idea of how complex the process is.
Using computer animation based on molecular research, we are now able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart the DNA double helix and cranking out a copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into 2 strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is 2 new DNA molecules.
So you see, it's a complex process. It takes a little bit of time to see how it works. Now, in addition to the helicase that unzips the DNA strand, there are 2 other really important molecules there, and they're called DNA ligase and DNA polymerase. Notice that all 3 of these, helicase, ligase, and polymerase, all end in ase, which means they are enzymes. DNA ligase is going to join the fragments together into a continuous chain, especially on that side that copies discontinuously. DNA polymerase is the main molecule that adds nucleotides to the chain and also proofreads and corrects improper base pairings. Every now and then there's a typographical error, and so DNA polymerase checks it and makes it and tries to correct everything that happens. And that way you end up with an identical strand of DNA for identical pieces of DNA for each cell in your body. The DNA polymerases also repair DNA that's damaged by radiation or toxic chemicals, things like that. It's really important that it works that way so that when you're getting copies of the DNA, that the mistakes that were made or the damage that has occurred can be corrected. This makes sure that all of your body cells, which we call somatic cells, carry the same genetic information and can perform the same tasks that need to be done by those cells of that organism. This shows the fact that the replication can start at many different sites and occur in both directions at the same time. So notice here's our parental DNA molecule, okay, and we're going to have these bubbles that form. One side gets copied one direction and the other side gets copied the other direction. And so basically you have the blue is the parental strand and the gray is the daughter strand like we saw before. And as the bubbles get larger from each origin, then they coalesce together so that you finally end up with 2 daughter DNA molecules that are identical to each other, half old and half new. This explains a little bit more about the 5' and 3' end. And so remember here, if we had an oxygen here at the tip of our pyramid, here's our number 1 carbon, 2 carbon, 3, 4, and 5. The phosphate is attached to the 5 carbon and the 3 carbon is attached to the previous phosphate in the previous nucleotide. So this is the 3' end, this is the 5' end, and the other side is exactly the opposite. Here's our oxygen here, and here's our 1, 2, 3, 4, and 5. Okay, so our 3 is going to be at the top on this side, and our 5 is going to be at the bottom on the other side. So as you're copying the DNA, you're going to add this nucleotide to here, and then you're going to come and add this one, and then you're going to add this one, and then you're going to add this one and continue going up the strand, this direction, when you're copying that side. If you're copying this side, then you have to start here and work your way backwards because you can only add it to the 5' end of the 5' end of the molecule. All right. Here, this shows you the DNA polymerase molecule. There are multiple molecules of DNA polymerase that are used in this whole process. And so once again, you have this strand synthesized continuously in the 3' to 5' direction, and we're adding from the 5' end there. And in this strand, it opens up, and you copy this part, and then it pulls out some more, and it copies this part, and then it pulls out some more and copies this part, and as it unzips more and more, it copies pieces at a time, one piece at a time, so in a discontinuous, and then here's our DNA ligase that's basically attaching those pieces together so that you end up with 2 full molecules of DNA. This video does a better job of explaining it so you can see what's happening rather than just listening to my words.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the 2 strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming 2 replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are 2 identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
So that shows you a little bit more about how that works. Okay? So last video, and this is the end of the notes for this part. This is the same video we saw before, not the first one that we saw, but with a little bit more detail about what's going on.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems. The separated strands are called 3' and 5', distinguished by the direction in which their component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
And that concludes the notes for part 2.